This podcast introduces epithelial tissue, and you were given a pink sheet with all the important things, the notes about epithelial tissue. So I'm going to go over this, draw a few pictures to help you understand what is on this side of the pink sheet. And then you also will receive or have received an ET glands pink sheet that talks about how we classify the different kinds of glands in the human body. And then the last thing we're going to use is in your lab pack on page 12. And on page 12, there's a chart here basically telling you the 8 specific kinds of epithelial tissue. It's going to give you the official abbreviation for each one, location in the body, and that location in the body correlates with the slides we're going to look at in lab this week. And then the functions for this specific kind of epithelial tissue. So let's get started with epithelial tissue. Basically, how are you going to identify it? And I'm going to give you some pointers and some drawings, and then we'll go through the pink sheet right here of notes. All right, so basically the first thing up here is epithelial tissue. Basically, if you got to break down the word, epi means above and thelial means to grow. So this first general type of tissue, and remember we have 4 general types of tissue, ET, CT, MT, and NT. And so epithelial tissue grows on the top surfaces, on the outside of the body. It covers any space in the inside of the body. It covers all internal organs, some kind of ET. And so basically that's where you're going to find it. It grows on the outside of the body or around air spaces in the inside of the body. So basically epithelial tissue, there's one thing you should know about it first. What color does it stain? So you can always identify it on slides. It stains basophilic. And so basophilic means the cytoplasm is going to stain some shade of purple. Light purple, darker purple, it just depends. Reddish purple, but it's going to have a purple tinge to it, okay? Now, basically we have 2 groups of epithelial tissue. We can divide all the different kinds of epithelial tissue into 2 major groups. One is called simple ET, which means there's just one layer of connected cells, and then stratified ET means that basically there has to be 2 or more layers of connected cells. All right, so I have them drawn on the board, and I'm going to give you a little introduction as to what they look like, okay? So over here, we're going to do the 4 specific types of simple ET, okay? And so here's the first one. It's called simple squamous ET, and it's abbreviated S as QET. And here it is. And so I think of squamous as squashed, like it's roadkill. It's been run over many times, and it's very thin cells. And so the nucleus right here is purple, and the nucleus basically is always flat and in the center of the cells. You can barely see it right here. So it's a very thin, like a stretched rubber band thin type of tissue, just one layer of cells. Now, the other thing about ET is that basically it always has to have something underneath it to support it, and we call this the basement membrane. And the basement membrane is basically sort of like super glue. It's going to stick the ET cells to the CT that is always underneath ET. There's no exception to that. And so basically it's like super glue, the basement membrane. So it will be underneath each kind of basically epithelial tissue. So here's the basement membrane, and I'll talk a little bit more about it when I get to another diagram. But here's simple squamous, think squashed, flattened, roadkill. Simple QBT is like a bracelet. They're nice cube, little square cells, and they have a nice round nucleus right in the middle. And so it never squashes down, that nucleus, no matter how much pressure you put on it. It's sort of like those bouncy balls, those hard balls you get out of like a gumball machine, that you hit it and it goes everywhere. And so that nucleus always is round and small, dead center in the cells. And then here's the basement membrane, and it sort of stains a pinky color, a clear color. And so it's really hard to see the basement membrane, but it's under this one layer of cells that are connected together, okay? The third kind is called simple columnar ET, and we abbreviate it SCOL-ET, simple columnar ET. And these are tall cells, very tall. They have an oval nucleus at the base of the cell. At the base means at the bottom. And then here's the basement membrane underneath it, right there. And so very tall, thin cells, and they always have an oval nucleus at the bottom third base of the cell. And our last type of simple ET, which is just one layer of connected cells, is called pseudostratified columnar ET, and it abbreviates PSTCOLET. And you are free to use abbreviations on a test, and you just have to know what they mean, of course. And so here are pseudostratified. It's sort of like a bunch of columnar cells that get squashed together, and the nucleus is still oval, but it sort of floats in the middle of the cell. And so some of the cells don't look perfect, like these cells down here, but some of them get squashed in here, and they're different shapes, but they always have an oval nucleus, but it's about halfway up in the cytoplasm, okay? So those are my four specific types of simple squamous ET. And here I have the basement membrane. What's it made out of? It's made out of glycoproteins. And so glyco is always going to mean starch. Glyco means starch, which is nothing more than a big sugar, okay? And it's made in the SER. Do you know what that is from 101? It's a smooth endoplasmic reticulum. And so it's going to put together the starch for this gooey stuff. And then it's made out of protein. And Golgi and the RER, the rough endoplasmic reticulum and the Golgi apparatus, they make the protein and they stick it to the starch, and this stuff then sticks to the base of the ET cells right down here. So what kind of tissue is going to be underneath CT? There's going to be right here some kind of CT is going to be under this ET always, okay? And so under here will be CT, some kind of CT under the basement membrane right here. So the basement membrane is an important concept. Can you see it in the slides? No, not always. Hard to see, but basically it's sticking the ET to the CT that is underneath it right there. And that's the function of the good old basement membrane. Now we have four more specific types of ET, and they're over here on the end of the board. They're called stratified ETs. Four specific types here. And so basically I drew little pictures of all of them. And how do you ID these? Well, you always want to look at the shape of the cells at the top, a very top layer of the cells. And so if it's stratified ET, you have to have two or more layers of cells all hooked together, okay? And so basically, here are the big four right here. So the first one's called stratified squamous ET, just like simple squamous, but stratified. So here are the cells right here, and they have a round nucleus underneath, but when they get to the top, when they get to the very top, then the cells start to die, and they get flattened and squashed, and then they have the typical squamous cell, a very flat cell with a flat nucleus, and there it would be. So stratified is always named for the shape of the cells up on the very top. And so stratified squamous, you're looking at it right here. This is your skin. Your epidermis is made out of stratified squamous ET. And of course, the orange right here is the basement membrane, and some kind of CT will be hooked to your skin. So you've got connective tissue right underneath this layer. If I can peel this back, there is definitely a layer of CT underneath it for support and for feeding it and controlling everything that ET will do. Then the second kind, specific kind of ET, is called stratified cube ET, and it only has 2 to 3 layers of cells. And so skin, stratified squamous ET, oh my gosh, you can have up to 100 layers of cells in just the epidermis right there. But the stratified cube and stratified columnar ET, both of these only have 3 layers max, okay? So here's stratified cube. They just look like simple cube, but there's 3 layers of them, like little bricks. And so they have an absolutely round nucleus right in the center of the cell, just like simple cube ET over here, right there, with the little simple ones all hooked together. But now I've got 3 layers stacked on top of each other like bricks, and the nucleus is round and dead center in the middle. And then stratified columnar ET, again, has just 3 layers, and the bottom 2 will have a round nucleus, but the top layer is what you always want to look at, and here they're very tall cells with an oval nucleus at the base of the cells right there, and so that's stratified columnar ET, okay? And then transitional ET. Transitional ET, basically, this is only found in the bladder, okay? In the bladder. And so basically, transitional ET has many layers, okay? And the cells underneath sort of look like cubes. And so they have a nice little round nucleus on the bottom layers. But then when you get up to the top, they become very fluffy and large right here. And so very large round nucleus. And some of them might have 2 nuclei in one cell. And that's called binucleate. And so basically very, very large cells up there. And what's going to be there will be your urine right here, your urine. So transitional is only in the bladder, only inside the bladder. That's where you're going to find it. And so it's the 4th specific type of ET right there. All right, so now that's a little introduction to all of the ETs. And so on your pink sheet right here, basically it says on the very bottom, it says criteria used to classify specific types of ET. So what can we use out of all of these 8 pictures to basically say, oh, this is simple cube versus simple squamous, or stratified squamous, or this is simple columnar versus stratified columnar. What are the things that I'm going to train you to use to look at in the 4 general types of tissue, okay? First of all, the first thing you always want to look at to be able to say this is some kind of ET is the number of cell layers present, okay? Is it simple or is it stratified, okay? Number of cell layers present. Number 2, overall cell shape. What does it look like? Does it look like a square? Does it look squashed? Does it look like a rectangle? What does it look like, okay? Nucleus shape. You're always going to look at the shape of the nucleus and where is the nucleus inside the cytoplasm. And so it's an oval shape and at the base, at the bottom. Round shape, dead center, right. And so you always want to look at the nucleus shape. And then the staining color of the cytoplasm. So all ET has purple cytoplasm. So is it sort of light color? Or is it going to be dark color? Is it going to be really dark? Or is it going to be light? Okay? And so that's something you can look at in all of these. It might be just very light color, and it might be darker in this one. Okay? Like that, a dark purple. So always look at the staining color of the cytoplasm. And then the last thing we can use to identify ET, some of the ETs will have cell surface modifications. And what does that mean? Well, I'm going to go over those in a minute, but cell surface modifications means something is attached on the top surface right here. And so basically, we could have cilia on some of these cells. We could have little cilia right here. And so that would be a cell surface modification. Little bitty hairs that move back and forth. We could have microvilli. And microvilli are cell membrane extensions like this. And so that's microvilli right there. And that's a cell surface modification. And so microvilli are going to increase the cell surface area so that you can absorb more food, more oxygen, whatever you want, it's going to absorb more of because it's just not flat and straight across. And then what other cell surface modification can you have? You can have flagella, long hair-like structures attached to a cell. And so basically you can have a big long hair like that. And that would be a flagellum. And that's just a long hair. And you've had all this in 101 biology, and that can move the cell. Flagellum can move a cell. And there's only one cell that actually has a flagellum, and only guys have it, and that would be the sperm tail. Right, the sperm cell is a simple QBT with a tail on it called the flagellum, right? So, and that makes it move. All right, and then there's one more cell surface modification for ET cells, and guys have this, only in guys. And so they have a kind of microvilli that actually helps feed sperm. And so basically the cell membrane will go up like this and make like, sort of like a long finger, like that. And that kind of cell surface modification is called the stereocilia, right there. Stereocilia, right there, stereocilia, and that helps feed sperm, right? So those are all listed in your notes, general characteristics, basically. So now we got the criteria for naming these types of cells, their specific names, and now we have general characteristics for all specific types. What do these all share in common? What do they all have in common? All 8 specific kinds of things. So these are called the general characteristics for ET. So number 1 is location in the body, okay? Location in the body means they're found either on all body surfaces, like your skin. They can line body cavities, spaces in the inside of the body. They make up all glands. Anytime you hear something is a gland, then that's made out of ET. And it's found on all outside surfaces of internal organs. So when I open you up and I look at all your organs, it will have a nice shiny layer of ET on it, giving it some shine, and that is always simple squamous ET. It goes around all the organs in the inside of the body and gives it shape. Number 2, all ET cells, the bottom layer of cells, or if it's simple, the bottom layer of cells makes the basement membrane. And so when you have stratified ET right here, the bottom layer of cells will make the basement membrane. So basement membrane will be made by these cells, and then it gets secreted out. It's going to come out and underneath the bottom layer of cells so that you can actually, it'll be made down here at the base of the cells, and then it's going to come out and become the basement membrane, right? So that's what that means. That's number 2. All right, number 3, CT is always found under ET. That is absolutely true. 4, all ET stains basophilic. So basically, a base pH is 8 to 14, like we did last week for histotechnique. And some shade of purple, basophilic cytoplasm, will be in all of your ETs. 5, high cell density. So the cells don't have any spaces between them, no. And so they are connected, one after the other. They're like sewn together. And so these simple ones basically are sewn together. And there, over here, they're going to be sewn together. So basically, that is high cell density. They're attached to one another. They're cell membranes. And so how do they stay together? Well, they actually have little filaments. They have little filaments that are going to go through the pores. So you've got little spaces in your cell membrane like this, little spaces. And these things are going to go through and they like thread through. So here would be a pore. And these things are called microfilament or myofilament, right there, okay? So micro or myofilament, right there. And so those are going to actually help attach all of your cells together. And so all of them, they'll have pores here, and the microfilaments will go through, and it's like sewing them together. Over here on the stratifieds, oh my gosh, they go this way, this way, this way, every which way to keep them together, right? And so they're all sewn together, absolutely, this way, every which way, so that they can hold together so that you don't melt down, right, exactly. So those are called myofilaments, and they're number 6 on your pink sheet. And number 7, cell surfaces. So all ET cells have a top surface and a bottom surface. Top, bottom surface. And so the top surface is called the apical. Apical means top surface. And then basal, here, right here, basal is the bottom surface of the cell. The bottom surface, right down here, right there, okay? And so they always have the 2 surfaces, top and bottom, apical, basal, or apex, basal, right, base. All right, so that's number 7. Now number 8 is all ETs. You notice there are no blood vessels or nerves, nothing in between these cells. No, the blood vessel nerves are going to be underneath in the connective tissue. So all ET gets fed and controlled by the nerves and the blood vessels underneath in the connective tissue, okay? So that is number 8. And number 9, organelles are polarized. What does this mean? There are certain organelles that are only found in certain locations in the cell, like this. Like mitochondria in simple columnar ET cells are only found on the bottom. And so polarized organelles means only in specific area of the cell. They're not found floating. They're not found floating all over. So the mitochondria can be only at the bottom, or you can have a lot of them up at the top if you have to bring a lot of stuff through. And so certain organelles are found only in certain places, like the nucleus. The nucleus isn't a true organelle, it's a structure, but basically the nucleus is always found in the middle of simple cube ET cells. So that's what number 9 is trying to tell you. And then number 10, ET cells can have cell surface modifications. This is number 10. And so here are the examples of cell surface modifications: cilia, flagellum, stereocilia, and microvilli. And so that would be number 10. And then number 11, the only ET cells that are not hooked together are your gametes, your sex cells, sperm and egg cells. Those are the only 2 that are not hooked together. They are single cells that are released from the ovary or testes. So now we just went through all the general characteristics that all of these 8 specific types share in common. And so we have just completed everything on this sheet right here, except for ET glands. Okay, now on page 12 from the Lab Pack 12, and on 13, it's an epithelial tissue chart or table. And basically on 13, it's empty. So that basically it's there for you to see if you can remember what it said on the other side. So it's being used as a study tool, essentially. But what is on the front side is basically here are the 8 specific types in the first column, the tissue name, specific tissue name, their abbreviation, which we already did on the board, okay? Location, basically. So for simple squamous ET, it's on the outside of every internal organ, okay? And what does it do? It gives it shape to the internal organs. So when you see the heart, it's gonna look heart-shaped. When you see kidney, it looks like a kidney bean shape. The brain, it's gonna have that shape. So it gives shape to all your organs. That's what simple squamous does. It's like a little thin plastic layer around the organ. Okay, simple QBT. Basically, lots of them. Lots are found in the kidneys and in glands. Anything called a gland and kidneys are loaded with simple cube ET. Simple cube ET are very good for support. They're very stable and they hold their shape well. So when you think of simple cube ET, you should think of bricks, okay? Bricks in a house and stacking so they give good support, they don't crush down, and so great shape to organs. Then simple columnar ET. Simple columnar ET are these tall, thin ones. And where do you find simple columnar ET? Large and small intestines. And what do they do? Basically, they're nice tall cells, so they're going to transport a lot of food through them. They can move a lot of liquids, and so they're for storage and absorption of lots of things, mainly food items, waste, etc. And then pseudostratified columnar ET, right above it, right here. Basically, it's going to be in your respiratory system. So inside your nose, inside your lungs, your trachea. And so basically it lines all of those. And what does it do? It moves air and it makes mucus. So this type of ET, very specific for the respiratory system. So it's going to move air and it will have cilia on it up there to help move the air and the mucus to help trap air and keep it moist. All right, then we go to the stratified squamous, the 4 over here. So stratified squamous ET, that's a nice easy one. It makes up the epidermis of the skin, and we will see a slide on that. And what's it for? Environmental protection, right? Among many other things. So when we get to the skin system, the integumentary system, we will discuss, like maybe 15 more functions for the epidermis, but basically stratified squamous ET is this specific tissue right out here, and it's protecting you from the environment. So that's good for right now. Then we have stratified QBT. These are in the ducts of glands, and so the ducts are like straws, and so they're going to suck out the secretion like saliva or mucus, whatever it might be. And so think of it as a straw, basically in a gland. And so it's going to give good support, again, good support for the straw, the duct. And then stratified columnar right here. Also, it does the same thing as stratified QBT. It's going to make the ducts of glands, bigger glands, and gives great support, these two together. And then the last type of specific ET, transitional ET, again, is only inside the bladder, urinary bladder. And what does it do? It allows for stretch so that your bladder is like a balloon. And so when it's empty, okay, great. And then as it fills with urine, then basically it allows it to stretch like a balloon. And so this type of ET, very stretchable. And so those are the specific functions for the 8 specific types of ET.